So with no further ado, I'll introduce Dr. Nathan Wiederhold. Nathan is Associate Professor at the University of Texas Health Science Center in San Antonio with us. He now directs the fungus testing lab and is doing a terrific job in that setting, and was going to talk to us about the epidemiology and resistance of Aspergillus and molds, particularly from a lab perspective as he's seen it.
I want to kind of make one quick statement. As Pete said on our session on Tuesday evening, we're in a chapel, so as it came up in our breakfast discussion, there are probably a lot of people in the audience glad that I'm not Mike Rinaldi giving this talk this morning. So what we're going to talk about is the epidemiology and resistance of Aspergillus as well as other molds. So we're going to start off with some very general comments that most of you in the audience are very much aware of. And that's that invasive mold infections are opportunistic infections. So we have to give them an opportunity in the host in order to cause disease. However, in those populations, they are associated with significant morbidity and mortality. Patient groups at highest risk include those with hematologic malignancies, those receiving stem cell transplants, as well as solid organ transplant recipients. And for many of these, we have limited treatment options. Many of the mucormycoses can be rather aggressive, although our options have increased recently for the treatment of these infections. But some that we still are lacking on include Fusarium and Scedosporium lumentospora species. And while our knowledge of fungal taxonomy is increasing, as evident by several of the talks that have been given this week, our diagnostic strategies as well as some of our treatment strategies are still somewhat lacking. So what we're going to do first and foremost is kind of go over some of the literature on epidemiology, and really going to focus on what came out of the Transnet as well as the Path Alliance studies to kind of set the stage and then go and talk about some specific invasive mold infections. So the first slide here is from the Transnet Organ Transplant Recipient Database. And so this collected cases of proven and probable invasive fungal infections from 15 U.S. centers from 2001 through 2006. And it was shown that invasive candidiasis was the most prevalent invasive fungal infections. This was followed by invasive aspergillosis, but there were some other molds as well that popped up. Some of the phaoid molds that we'll talk about a little bit later, some that were unspecified, we really couldn't tell exactly what the mold was, and then also mucormycosis, which we'll spend some time on a little bit later in the talk. Now, we took a subset of the Transnet solid organ patients and looked at those that had received lung transplants. Overall, there were 143 lung transplant recipients that developed invasive mold infections, so just over 12% within that period of time. The most common one, of course, was Aspergillus. Other molds, which are listed toward the bottom of the screen, many of those were the phaoid species. Also, Scedosporiums popped up, and that really wasn't seen in other solid organ transplants other than what was seen in the lungs. Overall, the invasive mold infections occurred about 11 months, a median of about 11 months post-transplant. Majority were late onset, and that's been a kind of a consistent thing that came out of the Transnet as well as the Path Alliance data. And for mucormycosis, the median onset was actually 26 months post-transplant. For the population that had received stem cell transplants, a somewhat different picture was observed. Instead of invasive candidiasis being the most prevalent invasive fungal infection, it was actually invasive aspergillosis. And mucormycosis was also higher, 8% here, 9% if you look at the incidence cohort, where in contrast in the solid organ transplant recipients, it was just over 2%. Other molds, those that were unspecified, and then also fusariosis was also seen, particularly within this cohort. Now, the invasive aspergillosis being the #1 cause of invasive fungal infection wasn't just limited to the Transnet data. was also seen in the Path Alliance group. So this was 23 medical centers in the US and Canada between a similar time frame, July of 2004 to September of 2007. And again, invasive aspergillosis was the most common invasive fungal infection, again, topping invasive candidiasis. Mucormycosis just over 7%, very similar to what was seen in the Transnet cohort of stem cell transplant recipients. And the Path Alliance also reported outcomes. And so for invasive candidiasis and invasive aspergillosis, majority of patients actually had a complete or partial response to therapy. In contrast, much lower response rates for patients who had mucormycosis as well as other molds, including fusariosis. The last thing I want to talk about in terms of epidemiology that came from the Transnet study is that of phaeohyphomycosis. This is often included in the other molds category. So these are the dematiaceous or dark molds that were causing infections within these patients. Overall, about 2.6% of invasive fungal infections in the Transnet cohort were due to phaeohyphomycosis. And although they were similarly divided between the solid organ transplant and the stem cell transplant recipients, there were some key differences. So for example, those who had received a stem cell transplant were more likely to have pulmonary infection. In contrast, if you had a solid organ transplant, it's much more of a cutaneous infection. Overall, 90-day mortality was also higher in the stem cell transplant cohort. The species that were commonly seen included Alternaria, Exophylla, Cladophyllophyra, Scopulariopsis, Curvularia, Phylumonium, and Xerohilum. And so, as you know, our treatment options against some of these is quite limited. So this is kind of what's been seen in the epidemiology studies, but it's important to remember that these studies, both the Transnet and the Path Alliance, they stopped enrolling patients really between 9 to 10 years ago into these studies. And so we've had a lot of changes within clinical mycology and the treatment of invasive fungal infections and invasive mold infections during that time. So that brings up the question about whether or not, while these data are very important and still very useful, whether or not they are still up to speed with what we're seeing right now. So what I'd like to do a little bit now is kind of switch gears and talk a little bit about the more common invasive mold infections that we're seeing both in Transnet and in the Path Alliance. The first one is aspergillosis. I want to talk about this because it was the most common invasive mold infection that has been reported within these at high risk populations. Overall, acute invasive aspergillosis has been estimated to be about 200,000 cases per year. Again, the leading cause of invasive mold infections in solid organ transplant recipients, lung transplant patients, those who receive stem cells. In fact, in the Transnet and the Path Alliance, this was the most common invasive fungal infection, not just invasive mold infection, but invasive fungal infection. And there's also been some estimates from David Denning's group that there are about 3 million people worldwide who have chronic pulmonary aspergillosis, and it's really seen in those who have some type of underlying lung disease such as tuberculosis and sarcoidosis. As you all know, aspergillus from a microbiology standpoint, these are ubiquitous molds. There are greater now than 200 species that have been described. Common ones that we see in humans include Aspergillus flavus, Terrius, Niger, and then the one that's most commonly seen is Aspergillus fumigatus. The primary organ where we see invasive disease is in the lungs, and that's because these organisms get into us as we inhale the conidia. We do have expanded options now for the treatment of invasive aspergillosis, specifically the treatment of infections caused by Aspergillus fumigatus. So here we have some MIC data that comes from a query of the antifungal susceptibility database in the fungus testing lab. This was between 2001 and 2013, where we had over 1,100 MICs for itraconazole, over 1,700 MICs for voriconazole, and just over 800 for posaconazole. Overall, we still have very good activity for these three agents. We're starting to collect data now for isavuconazole as well, and these data seem to hold true. However, we did see some resistance as well, as shown in the graphs at the bottom of this slide, anywhere ranging between 2.5% to just over 5%. Now, I do want to caution you and say that I think these rates of resistance might be a little bit high. Most of these isolates come from the United States, but because this was a query of our database and we did not go and confirm the species identity as Aspergillus fumigatus, many of these could be a cryptic species for which the azole resistance or azole susceptibility is already somewhat reduced. But it does help lead into the next slide, which is something that start to gather attention in medical mycology, and that's azole resistance in Aspergillus. We know that this is due to point mutations within CYP51A, which is the gene that encodes the 14-alpha demethylase, the target of the azoles. This has been observed both in clinical and laboratory isolates. And we also know that the position of the point mutation determines azole resistance. Some point mutations lead to amino acid changes that cause pan-azole resistance. Others seem to affect primarily posaconazole and itraconazole, while others affect voriconazole and izabuconazole. Now, historically, what we've seen is that with chronic azole exposure in patients, these are the ones that seem to be at highest risk for the developing microbiologic and clinical failure. Where we really see this has been reported significantly is in patients with chronic pulmonary aspergillosis, really seen in high rates in Manchester in the United Kingdom. What was also start to capture the attention of the mycology community is that of environmental exposure of Aspergillus fumigatus to the azoles, and if that can actually lead to resistance, that can be important clinically. This was first recognized in patients in the Netherlands where they had cases of azole-resistant invasive aspergillosis in patients without prior azole exposure. Subsequent investigations into the indoor environment of the hospitals and areas in direct proximity to the medical center, as well as fields where azole fungicides were used, where they were able to find some Aspergillus fumigatus isolates that were indeed resistant to the azoles. Now, the mechanisms that you see with environmental exposure are somewhat similar to what you see with clinical exposure. There seem to be point mutations within the CYP51A gene that lead to amino acid changes. But what we also see in these environmental isolates is a tandem repeat of base pairs, either usually of 34 base pairs in length or 46, which are the most common. And these are occurring within the promoter region of the CYP51A gene. Now, this has now become a worldwide issue. This was a table that was adapted from a recent review article published in the Journal of Fungi, which kind of shows different continents and countries and whether or not they've seen clinical isolates as well as those that have mechanisms of resistance associated with environmental exposure. And as you can see, found it in numerous countries within Europe. It has now been documented both in North and South America, Africa, at least with environmental isolates so far, as well as in several countries in Asia, as well as in Australia. So this slide here kind of shows our experience in the fungus testing line. We published this in January of this year. So out of a collection of about 200 isolates that we had subcultured, again, from our stocks, we identified 26 isolates that we confirmed truly to be Aspergillus fumigatus and that had high azole MICs. Of these, in this collection of 26 isolates, we had 4 isolates with mutations that were associated with environmental exposure to the azoles. 2 of them that had the TR34L90H, which gives basically pan-resistance to the azoles, and then also one that had the 46 base pair tandem repeat within the promoter region, which seems to affect voriconazole and izabuconazole more than posaconazole. One of the isolates, one of the TR46 isolates, it was actually sent to us back in 2008 from a patient with documented, proven invasive aspergillosis in Arizona, and actually predated the first description of this particular mutation in the Netherlands in 2009. So again, we're talking about, we've heard a lot about emerging infections. Well, a lot of these really aren't emerging infections. It's our knowledge that's really emerging, our understanding that's really increasing. Now, and again, about the same time that we were publishing our results, Jose Vasquez, who's here in the audience, they published a case report in Antimicrobial Agents and Chemotherapy describing a patient, I believe from the Midwest United States, who also had invasive aspergillosis, and that isolate was resistant to voriconazole due to the TR46 mechanism of resistance. One other thing I want to point out is that in our collection of these 26 isolates, we had 6 isolates confirmed to be Aspergillus fumigatus, definitely had elevated azole MICs, but were without a CYP51A mutation. Now, there are other things that have been described that might lead to azole resistance in Aspergillus fumigatus. Some have said that there could be an overexpression of C51B. There's actually a B component to CYP51. It doesn't seem to be really associated, though, with ergosterol production, so it's really the role of that specific gene in azole resistance is unclear. Efflux pumps have also been proposed, similar to what we see in Canada species. In Aspergillus fumigatus, there are about 50 ATP binding cassette family transporters and 300 major facilitator genes that are predicted in Aspergillus fumigatus. And in a laboratory settings, we know that overexpression of several of these, including one that's associated with biofilm resistance in Aspergillus fumigatus, can lead to azole resistance, at least in vitro. Another mechanism that's been proposed is cholesterol import, and so Aspergillus fumigatus can apparently import exogenous cholesterol in order to replace the ergosterol. Since we're inhibiting the biosynthesis of ergosterol with the azoles, it can import exogenous cholesterol and use that, replace that in its cell membrane. There's also been a gain-of-function mutation within HAP-E, which is a cat-binding transcription factor complex subunit. And when you have that gain-of-function mutation within the HAP-E complex, you can have binding of the, to the cat box in the promoter region, and this can actually induce the expression of CYP51A. Now, it's important to realize, though, that while these have definitely been shown in vitro and in laboratory settings to result in azole resistance, we really don't have a good understanding of the clinical role this might have in microbiological failure and then clinical failure within our patients. And the reason that this might be is because we're really looking at the low-hanging fruit. It's very easy to sequence and look for point mutations within CYP51A, but at least from a clinical laboratory standpoint, doing these other investigations is not something that's at least feasible from a clinical microbiology standpoint. Taking a step back, it's also important to remember that in addition to acquired resistance, we can see intrinsic resistance. So again, we're seeing an increase in the number of infections due to non-Aspergillus fumigatus isolates. And again, these aren't really emerging pathogens. They've been there. It's our understanding and our recognition of them that's really emerging. So in terms of the leading causes of invasive aspergillosis, Aspergillus fumigatus still at most institutions is the leading cause, followed by Flavus, Terrius, as well as Niger. But now with the molecular advent that we've seen in clinical microbiology and mycology, we're starting to recognize cryptic or sibling species. And what that means is it's really difficult to differentiate between them by classical means, by phenotypic and morphologic characteristics alone. In the Transnet study, it was reported that about 11% of the Aspergillus species were actually considered to be cryptic. Those included Aspergillus lentulus, Aspergillus udogawi, formerly known as Neosartoria udogawi, Aspergillus tubangensis, as well as Aspergillus callidus. In a study that came out of Spain, another epidemiology and surveillance study, they reported about 14.5% of cryptic species among their 323 Aspergillus isolates. Again, common ones that were seen were Aspergillus lentulus, Aspergillus tubangensis, and then also Kaleidoeustis. Now, within Aspergillus section Fumigati, which is also referred to as the Aspergillus fumigatus species complex, it's now known that there are at least 51 phylogenetically distinct species. We're not really able to discriminate between them very well based on morphology because it's been stated they have basically unstable morphology with different exposures to environmental stresses, including antifungal drugs. The morphology of these can change, and we're really not able just to look under a microscope and really very easily classify them correctly to the species level. It now requires either molecular or proteomic means, either by DNA sequence analysis of the beta-tubulin and calmodulin genes, as well as MALDI-TOF as an emerging technology for this purpose. Now, of the 51 phylogenetically distinct species within section Fumigati, 15 at least have been reported to cause disease in humans. Those that we've always known as Aspergillus species, and then those that were previously called Neosartoria. Now, this is important because many of these cryptic species within section Fumigati actually have reduced azole susceptibility, as shown in the slide here. The infections that they've been reported with in individual case reports include invasive pulmonary aspergillosis, osteomyelitis, peritonitis, as well as cerebral aspergillosis. Now, I've talked a lot about nomenclature change and changes in taxonomy. I do have some good news, and that's that Aspergillus fumigatus is not changing its name. And so the International Commission on Penicillium and Aspergillosis took a vote on this back in 2012. And they basically said that the name Aspergillus is going to be maintained with some exceptions, although I don't think these are really clinically relevant. Those include the Neosartoria when recovered from canned products. I don't know the last time you actually opened a can and found a patient. The Eurotium when it's from salted fish, because apparently the ascospores are heat resistant. And then Emericella nigilans when the expression of genes during sexual development is examined. If you want to look specifically at the 5 options they considered when they voted to keep Aspergillus, you can look at this website. It also has a fairly up-to-date list of new names for both Aspergillus as well as Penicillium. Now, in addition to section Fumigati, there are several other sections that we have to consider. Flavi, Nigilantes, Nigri, Terrii, Usti, as well as versicoloris. And within each of these are cryptic or emerging species which can have reduced or variable susceptibility to different drugs, including amphotericin B. Now, this is definitely not a comprehensive list, but some of the ones that we've at least seen over the last couple of years come through the fungus testing lab. Switching gears for mucormycosis, the prevalence was much lower than what we saw with invasive aspergillosis, both in the Transnet cohorts of patients as well as in the Path Alliance. Individual studies that have been reported out of Johns Hopkins and an autopsy study out of UT MD Anderson from Dimitris Kotnianis and Russ Lewis, which showed that their prevalence was about 11.5%. Population studies have reported that overall in Spain, France, and California, cases per million population per year range anywhere from 0.4 to 1.7 cases. At MD Anderson, they have reported an increase, at least as seen in autopsy patients, in mucormycosis, and a slight decrease in invasive aspergillosis. However, as they state in this article, the number of autopsies that are now being done is far less than what had previously been done. Causative agents of mucormycosis, there are several different genera listed here, and some of the more common species that you can see causing infection in humans. But the one that really causes the majority of infections in the United States is that of Rhizopus oryzus, or also known in the literature as Rhizopus oryzae. Within the Path Alliance stem cell transplant cohort, it was responsible for about 55% of mucormycosis. It can be very aggressive, associated with high morbidity and mortality, especially in your immunocompromised population in which you can't correct the underlying immunodeficiency. In the past, it's been very difficult to treat, although our options are now increasing. Now, one of the things that we've started to become more interested in, at least in the fungus testing lab, is the difference between Rhizopus oryzus variety oryzus and Rhizopus oryzus variety delamar. So Abi and colleagues back in 2007 said that these are actually separate species. Phylogenetically, they're distinct by looking at both ITS alone as well as by multi-locus analysis. This has been in agreement with other studies that have looked at this. There are phenotypic differences. Rhizopus oryzus is able to produce lactic acid, whereas Rhizopus delamar is not because it lacks the LDHA gene. But in an article coming out of the CBS in the Netherlands, they actually made the argument that no, these are not separate species, but in fact, they're just varieties of Rhizopus oryzus. Now, some of you in the audience might be thinking, well, what does this have anything to do with the patients that I treat? Does this make any difference clinically? Well, there's another phenotypic characteristic that we've started to see in some of our isolates, and that's of susceptibility to the azoles. And so this table here and the graphs on the slide show our recent experience that was published as a study we did in collaboration with Ashraf Ibrahim at Harbor UCLA, in which we were looking at one of the Viamet investigational agents, VT1161. And against Rhizopus oryzus, it actually had fairly good MICs, ranging anywhere between 0.25 to 1 microgram per mil. In the animal model, in which the mice were infected with a Rhizopus oryzus variety oryzus strain, Viamet agent also resulted in significant improvements in survival and significant reductions both within the fungal burden within the lungs and the brain. One thing, though, we noticed, and unfortunately we didn't have the chance to test this in the animal model, is that when we looked at isolates that were of the variety delamar, the MICs were significantly higher, 8 to greater than 32 micrograms per mil. Now this, again, we don't know the clinical significance of this, and we don't know if those higher MICs would have translated into worse outcomes in the mouse model because, well, we just didn't test it, unfortunately. But this doesn't seem to be specific to the Viamet agent. The table at the bottom kind of shows a quick snapshot of geometric mean MICs for 60 Rhizopus oryzus isolates that we received over about the last 10 months in the fungus testing lab, evenly distributed between 31 variety oryzus and 29 variety delamar. Amphotericin B didn't seem to be affected, but the geometric mean for both itraconazole and posaconazole were significantly increased in isolates that were of the variety delamar. Again, we really don't know if this translates into anything that is clinically meaningful in our patients, but it's something that we've started to become interested in and starting to gather more data with. One of the other invasive mold infections that's important, at least specifically in invasive lung transplant recipients, is that of scutosporiosis. As you know, this is an invasive infection primarily in immunocompromised patients. You can cause breakthrough infections in those who are either persistently neutropenic or lymphopenic. Very similar to aspergillosis, it enters our body by the sinopulmonary route, and can be difficult to distinguish from invasive aspergillosis in some cases. It can also be a common colonizer in cystic fibrosis patients, and it can cause infections in survivors of